And so, when we talk about, the emergence of life, we're going to talk about emergence at sort of multiple levels. The emergence of consciousness. You've got the emergence of life. This is a term that gets used quite a bit in philosophy and elsewhere. Can you define for people exactly what you mean by emergence? When we say something's an emergent phenomenon, what exactly does that mean?
Well, one of the problems is the term has been used, as you say, so diversely. It's been used to talk about almost magic, how something that didn't exist, a rabbit coming out of the hat. To some extent, we have that feeling when we think about, for example, the origins of life. There was nothing like it there before. It's like pulling a rabbit out of the hat. But what we know about magic is it's all trickery. It's just how things get organized and how the causality is not obvious from the start, but shows up eventually. So emergence, first of all, is not magic. But what we do want to explain is how certain kinds of properties of things show up in the world when they didn't exist before that. The case of life that we've just started this conversation on maybe has to do with something like normativity, value, the fact that there can be good and bad chemistry. There's not good and bad chemistry in a cosmic sense, but for every bacterium and even for every virus, there is good and bad chemistry. We can talk about destroying, killing viruses. We've been in the process of trying to deal with how to get rid of viruses. It's bad for viruses, what we're doing. Having human beings that pass this stuff on by coughing and sneezing at each other is good for the viruses. That normativity appeared on the Earth somewhere around 3.8 billion years ago. That looks like rabbit coming out of the hat. We use the term emergence to talk about that. What I've tried to do is to say, no, emergence has a structure. It's not just magic. It's not just unguessable, impossible things suddenly showing up. It has a structure. And what is that structure? I've tried to identify it in the case of life. I've tried to say, look, what we know is the second law of thermodynamics has certain properties. But if you push things in the second law of thermodynamics far enough, you get self-organizing processes. I've called them morphodynamic processes, that is, form-generating processes, regularity-generating processes. And that they emerge in thermodynamic systems when energy production systems and dissipated systems are balanced to each other in certain ways. And what happens is you get processes that seem to invert the logic that was happening before that. As you invert superficially and locally what looks like what the second law of thermodynamics should have produced. The whirlpool is more organized. The question is there another step like that going to life? And what I tried to describe just a few minutes ago was how also juxtaposing morphodynamic processes, self-organizing processes, with respect to each other in such a way that they complement each other, also produces another reversal. And that is the reversal of these processes that tend to undermine themselves. Life doesn't tend to undermine itself. These are reversals. And so it has this sort of rabbit coming out of the hat appearance. But what I've tried to say is that, no, if we actually look at how this actually happens, there's a physical story to be told. It's not magic. But it also doesn't mean that it's completely reducible. And one of the ways that people have used the term emergence is to talk about it's not reductionism. Reductionism says that everything that's going on is just the bumping in of atoms bumping into each other, atoms in the void and moving around and bumping into each other and sticking with each other, affecting each other, and so on. Life is not just that. What we're talking about is something in addition. Now, and here's the interesting part, and this is where we began this discussion. What's being added are new kinds of constraints, new kinds of preventions, new kinds of absences. This is what information is about. Information is about what it's not. So what's actually happening is, no, we're not adding new kinds of laws of physics. We're not talking about adding new kinds of atoms, new kinds of materials. They could all have been explained with chemistry. In fact, organic chemistry can be explained chemically. How the organic molecules get generated is also a chemical process, but it's a specially constrained chemical process. So that I think of emergence as the very fact that new information, new constraints can always be created and can be destroyed. But when certain kinds of constraints are produced, the kind of constraints that I call teleodynamic constraints, that is the kind of constraints that we see in living processes that have an end, that tend to maintain themselves. Once you have that, now you have a system that can evolve, can get more complex. And in that respect, what I mean by getting more complex is you're adding more complex kinds of constraints. Our brain is not doing anything that's not possible chemically or physically, but it's doing it in a very uniquely constrained, in a complex, constrained way. It's adding new constraints. Constraints can be added. New kinds of prevention can be added to the world. And what's added is not more stuff, it's more constraint. And I think the reason that we've had trouble thinking about it is we don't like to think about absences being added. Absences seems like something that's being taken away. We don't think about complexity, increasing complexity, as being more reductions. In fact, I think that we have to begin thinking about complexity in terms of prevention, of absences.
Yeah, so information, when there's more information, you know more. That's how we sort of think about it in very basic terms. But what you're saying is information is a reduction in uncertainty. And so when you add more constraints to a system, you add more information to the system. But what you're really doing is sort of preventing it from taking on many other states.
Exactly.
And when we think about information in biophysical terms, the first thing that people probably think about is something like the genome, DNA or RNA. And, the sort of cartoon explanation of DNA is it's a blueprint for the organism. It contains the instructions for the organism. So on the one hand, you can have information encoded in the genome that will be expressed as development proceeds. On the other hand, much of stuff in cellular life isn't actually directly encoded by the genome. There's a lot of these sort of self-assembling molecules that do stuff that doesn't strictly depend on what's encoded in the genome. So in thinking about, the emergence of life and the complexification of life, if life is end-directed in the sense that it's wanting to reproduce itself, where does that end come from? Can it be encoded directly in the genome, or is it sort of coming from somewhere else?
Well, so the first point is that it had to emerge initially, right? The origins of life is actually about the origins of end-directedness. And the end is to maintain the ability to have end-directedness. So it's a circle in that respect. That is, I'm producing something to make it possible to produce something. That is the case for all life. In fact, that's you might say, the beginning of what we want to call self. And this is why it's going to expand well beyond the origins of life, because what we really want to explain is how self comes into the world. And that's, of course, what we want to explain in terms of us and in terms of what brains do, what consciousness is about, at the highest level. But if we don't understand the nature of self, we can't even get started. And so part of what I wanted to do in this work is to say, okay, let's try to get the most straightforward, simplest understanding of how self comes into the world. And the way it does is that it now constrains what can happen so that self doesn't disappear. If it gets damaged, it repairs itself. I actually think of reproduction as just a sort of variant of self-repair. That if I can repair myself, put myself back together again, that means I can also probably put together a replica of me. And so when I see the origins of DNA, I actually think of DNA as ways of offloading and remembering how it's done so that it doesn't have to be done dynamically, so that I can have a kind of a written base that is a structure. I can turn these constraints, which are just constraints on dynamics, limitations of how things happen, also structure represents constraint. And so the structure of a DNA molecule actually encodes constraints. So what we want to say is that DNA molecule, yes, it produces proteins by this complicated process, as well as other things like RNA molecules that themselves can regulate DNA molecules and regulate proteins. But in this process, what's happening is it produces some and not others. The number of possible proteins of 100 amino acids long is astronomical. Life produces a tiny, tiny fraction of possible proteins. And in this respect, what we can say is that DNA is a way of encoding and remembering the constraints by taking what were dynamical possibilities of possible interactions that could take place and offloading that information onto the constraint of a structure, the structure of a molecule. Now, what's interesting about this process is that as soon as you do that, you have something that's more durable than dynamical processes like the actual interactions of proteins in a cell. The durable thing can stay there because it's easily remembered. The power of DNA, in part, is the fact that it's a very robust molecule. It's hard to mess it up. And as a result, it's a great memory system. RNA molecules are not so good. They're easy to break up. But because of that, RNA molecules do a lot more regulation. But now here's the thing. I like to call this you've all heard of the selfish gene story. I like to think about this as the lazy gene story. If processes tend to happen spontaneously in the world, once you've got a bunch of molecules sitting out there, and just because of their shape and the way they interact, they spontaneously produce structures. They fall together in the way that snow crystals simply fall together in regular shapes because of the very nature of water molecules and their geometry. Those are things that, if that happens spontaneously, all DNA has to do is to remember the conditions that make it likely to happen. DNA doesn't have to do the work of putting things together. It just has to constrain the boundary conditions that make these self-organizing processes happen. One of my favorite examples of a self-organizing process like this is the Fibonacci series. Fibonacci series is a series of numbers: 1, 2, 3, 5. 3 plus 5 is 8. 8 plus 5 is 13, so on and so forth. Just adding the last two numbers gives you the Fibonacci series. The Fibonacci series is really remarkable because what it means is that the relative distance between any two adjacent sequences is basically the same. And this is what's produced also what somebody calls the golden mean, the golden section ratio, the thing that makes spirals regular as they grow out. Well, one of the most interesting spirals in life is what we call spiral phyllotaxis. What I mean by this is looking at the surface of a sunflower, for example. You see that the seeds are all organized in interesting kind of spiral patterns. In fact, the spiral of the sunflower has to do with two interlocking spirals that curve in opposite directions. And the number of curves of those spirals, curving to the right versus curving to the left, and in a sense, crossing over each other like this, turn out to be adjacent Fibonacci numbers. So that there's some, like in, for example, pine cones, one of the curves is a 5, and the other curve is typically an 8. There's 8 in one direction, 5 in the other direction, and they fit with each other. However, you can get much higher order relationships in things like sunflowers and things. It turns out that many, many plants are organized this way. Now, there's a real advantage to this organization because it also keeps, if you've got a stalk and a bunch of leaves, it keeps the leaves maximally from interfering with each other. If you want to have a really successful way of capturing sunlight, you want to have your leaves dispersed in such a way that they're minimally out of each other's way. The Fibonacci series allows that to happen. It's a very, very useful series. Well, why does it happen in the surface of a sunflower? It turns out that we can produce the same series by dropping droplets that will stay in a particular size and won't fit with each other into a bowl. They'll begin to form a Fibonacci spiral. We can now show this on an electrically charged surface, that little iron filings will organize themselves into successive Fibonacci spirals. These things happen spontaneously precisely because they're pushing each other out of position in just such a way that new parts added will have a new position and will be maximally out of position of the last ones. The Fibonacci relationship is very, very precise, but the genes don't have to have the information that says I need to produce 5 in this direction and 8 in that direction. All the genes have to do is to set up parameters in which they're diffusing elements that diffuse out and don't allow certain things to grow or show up until the concentrations are just right. So by setting the boundary conditions, the genes don't have to do this. My point of calling this the lazy gene hypothesis, that means that if by accident these kinds of regularities just happen to show up in life, the genes don't have to make it anymore. They can now just sort of back up and only do the boundary conditions that are necessary. The reason that we can have a genome that's in our body not much larger than the mouse genome, and yet build a body that's vastly more complex, build a brain that's vastly more complex, even though we have essentially the same number of genes for building our brain as the mouse does to build its brain, is that a lot of those processes self-organize, and the genes have just taken advantage of that. So it's an optimization process. And this is one of the reasons why bodies are even possible, because the genes have also, as I mentioned before, the constraints can be offloaded onto genes, but the constraints in genes can be offloaded onto these self-organizing processes. And this makes it possible to build bodies of incredible complexity.
Yeah, I was actually thinking that as you were speaking. How is it that you can have, whatever it is, 20,000 genes to create the complexity of a human body, and that's comparable or even sometimes less than organisms that look much less complex than us? And what you're saying is it has to do with the fact that the genes aren't directly encoding like every single thing that needs to happen in every single cell. They're just creating the boundary conditions, the general boundary conditions inside of which all of these self-assembling processes will just happen within the constraints of those conditions.
Right. And if you think about it, what we mean by boundary conditions are constraints on possible interactions. So this is again an example of building constraints upon constraints upon constraints. This is how complexity builds. But now, I always tell my students in my neuroscience classes, the number of genes that are necessary to build a mouse brain is roughly the size of the last digit of your little finger. And the numbers of genes, the corresponding genes to build your brain and my brain, which is roughly the size of a pineapple, are essentially the same. And yet the information in just the structure of a brain the size of a pineapple, in which every neuron is connected to every other neuron by at least 1,000 other connections, in both